Ms. Gent just got here just this semester. Marlene Blankenbuehler, she's been doing this for a while, and she's particularly good, or has a particularly good knowledge of all the diversity of programs and requirements, and can direct you to an advisor if you want to discuss with a particular advisor for some different areas. Anyway, some things here about doing better in the course.
I apologize, I just walked in. What's that for?
Oh, this is if you're thinking about alternate plans, career plans other than what you're into right now. Okay. And for that matter, I almost always tell all my advisees, pharmacy advisees, when they come in first semester, you may have your heart set on one thing, and then that's good. I mean, you want to be very interested in that. But you may want to think about an alternate plan too. You know, there may be alternate directions that you could go. Again, even still in the healthcare profession that might be a little bit different, but you can keep your eyes kind of open to those possibilities as well. And so if you do want to discuss with somebody about alternate plans, these are particularly good individuals to help you with those kind of decisions. Okay, and now we're ready to move on to our subject matter for today.
So I didn't miss anything really, did I? Cool.
What we're doing today, again, is overview of muscle tissue organization. We're going to start talking about muscle contraction as well. We're a little bit behind, so I'll have to catch us up at some point later, but I don't want to go through this too fast because some of this is complex. And our first topic is thinking about how muscles eventually develop and how they then grow as we get older in life, as we get bigger and your muscles get bigger, either as we age or ultimately maybe as we do exercises that are designed to build muscle size. Okay, first of all, let's think about how muscle fibers, that is muscle cells, develop. And we have, maybe there's a chalk here, have a couple of cells that I want to mention here about how muscle cells develop. The first one is called a myoblast. This is an embryonic stem cell. Myoblasts are embryonic stem cells. We find those in embryos or fetuses before an individual is born. And they make a muscle fiber by fusing together. That is, they join together, they fuse together. It makes a cell called a syncytium. Syn means together, or syncytal. Cytal means cell, means fused cell. They fuse together, and that's what makes skeletal muscle cells multinucleate. Is you have several of these fused together, and you get a very large cell that has lots of nuclei, and that makes a skeletal muscle cell. So this is one type of stem cell. Those disappear past birth or past neonatal time periods, and the only remaining stem cells that are left then are called satellite cells. There's not very many of these left. Skeletal muscle cells, once they're formed, they don't reproduce. Once you get an adult, a formed skeletal muscle cell, it does not reproduce. In fact, for the most part, you have the same number of skeletal muscle cells now as you did when you were born. Your number of muscle fibers, muscle cells really hasn't changed between the day of your birth and now. Very, very little. There are a few of these satellite cells, and we maybe see it. Here's an example. Here is a satellite cell as well. Satellite cells do persist in adulthood. Those are stem cells. And they do have limited capacity to fuse together to make a few new skeletal muscle cells, or they can fuse and repair damaged skeletal muscle cells. But that's pretty limited. That's kind of good and bad. It means there's almost no cancers of muscle tissue because they don't reproduce. Muscle cancers are quite rare. That doesn't happen very much. On the other hand, it means recovery or replacement or regeneration of damaged muscle tissue is almost non-existent. So when someone gets a severely damaged muscle tissue, even a physical trauma where a big chunk of it gets cut away or damaged, it doesn't really grow back very much at all.
So if it never grows back, how come it gets bigger, like when you work out or whatever?
And that is our next topic coming up right up here. So how does it get bigger? And how do we have the same number of cells now as we were born? How do we get bigger muscle cells? They actually grow. Your whole muscles grow not by getting new cells, but the individual cells get bigger. So as a person ages from an infant to an adult and the muscles get larger, It's not addition of new muscle cells, not replication of new muscle cells. The actual muscle cells, the muscle fibers get bigger. They often get larger in diameter particularly. And when a person, say, works out, does exercises designed to increase muscle size or muscle strength, what happens is, again, very little, if any, new muscle cells, but what you do get are larger muscle cells. The actual diameter of the muscles gets larger.
So that's why certain people can only get so big, even if they work out every day.
Yes, there's a genetic link to a lot of the things that we have in our bodies, and that is definitely one. Yeah, that we have limits, and we'll also talk about slow and fast twitch fibers, and that's also important, that same concept. We'll get to that on Friday, or maybe next Monday, I'm not sure which. And that's genetically determined, at least in part as well.
So I got one more. I used to work out a lot, so this is kind of interesting. So when someone takes anabolic steroids, how does that force them to become so big? Or just makes them grow to the maximum?
I don't know the mechanisms of that particularly.
Okay.
Other than, ana or anabid means to climb up or to go up. It means to get bigger in effect. So that's basically building or enlarging kind of a drug would be. But so I think is what it makes the cells bigger. Now how the molecular action on that works, don't know for sure.
Okay, so your muscles don't grow like your bones, like your bones kind of have like a cutoff date. I'm sure that they have like a capacity that they can get bigger too, but it's not determined by your age.
Not as much. You know, a person my age, if I was to work out with weight building activities, exercises, I could get larger muscles to a certain degree. But as Josh said, we're all somewhat limited by our genetics. I'm never going to look like Arnold Schwarzenegger. It's never going to happen. But get a little bit bigger. Or likewise, if I became I'm fairly active now. It's just more aerobic stuff. If I became very inactive, I would have a smaller muscle mass. Or if I had an injury, I'd have much smaller muscle mass. 2 years ago, I tore a knee meniscus in my left knee. And I had a big chunk of cartilage broke off and locked up in the hinge. It's a couple months before I could get the surgery on it. Darn insurance here was a problem about that. But anyway, it meant that I couldn't move my left leg for about a month and a half or 2 months there. And my left leg got like half the size of my right leg. It was amazing. It really shrunk up. And then eventually got larger again. That wasn't a change in the muscle fiber number. It was just the muscle fiber size because of use and disuse. And we'll talk more about exercise physiology and muscle physiology, especially on Friday. So we'll come back to that topic a bit more. We'll think about why marathon runners and sprinters look very different. There's a reason for that. Okay, let's go to some more terminology associated with our muscle fibers, that is, our muscle cells. We're going to talk about muscle contraction. That's what really we're leading up to, but we have to understand some of the terms before we can talk about how muscles contract. So as we go through this, I'm going to try to define all these different terms. First of all, what is our sarcolemma? Sarcolemma is the same thing as plasma membrane. It's the plasma membrane of a muscle cell, aka muscle fiber. We see that here. This is one entire muscle fiber, one entire muscle cell. The bluish area on the outside is the sarcolemma. By the way, you often see sarco with muscles or myo with muscles. Myo means muscles, abbreviation means muscles. Sarco means fleshy, as in flesh or meaty. You know, muscle. Meat is made up of skeletal muscle for the most part. Sarcolemma, thus, is the plasma membrane. We give it a special name because it's got some special properties, and we'll come to that here in a few minutes. Or maybe we'll get to it. We'll maybe get to it on Wednesday. I'm not sure if we'll get to that one or not. We'll get to some animations here a little bit as well. We'll see. Oops, I just clicked the wrong thing there. Try back on this one. Transverse tubules, aka T-tubules, is a short version of that. These represent these bluish tubes that you see extending down from the sarcolemma. So they are tubes and basically little indentations from this outer sarcolemma down deep into the muscle. And they're highly branched when they get down in there. So there are these tubes, these channels that extend down from the surface. The reason why we have these is these are big cells. to rely on diffusion of materials from that outside to the deepest parts of the cells would take a while. And muscle cells tend to react quickly. They start contracting quickly. They stop contracting quickly. They need a lot of energy. They need a lot of calcium. They need a lot of different things. And so it's very important to deliver materials down into there. And it's very important to, for events that start up at the surface of the cell to go down deeply into the cell. And that's what those T-tubules, transverse tubules do. We'll talk more about that in a little bit. So I know I haven't given you much to go on for T-tubules, transverse tubules, but we'll talk more about those as we get into contraction. See how all of these fit in.
So they carry stuff down to the sarcolemma?
Yes, in part, and also I've said something, events at the surface continue down. Sometimes it's electrical charge. So if the membrane becomes charged, they will carry that charge deep and down the membrane, and that charge is actually going to trigger contraction. But it can trigger contraction throughout the whole muscle cell, more or less, muscle fiber at once.
So it could be like it could be like nutrients too, though, couldn't it?
Could be, yes, yeah. Could be nutrients or oxygen or anything like that, yeah. Or removal of waste, whatever. It's kind of a way of increasing surface area of the whole plasma membrane, aka sarcolemma. Sarcoplasm is the same thing as cytoplasm. So again, we have a fancy word for the special cytoplasm inside of skeletal muscle cells. Skeletal muscle cells are organized differently than most cells. They have certain physiologic properties different than most cells. That's why we have some of these different words that mean the same thing. And like it or not, this is the way that names are done for skeletal muscle cells. So if a person becomes a healthcare worker or a researcher involved with this kind of tissue, this is the names that's going to be used. Some things that could be in the sarcoplasm that are important. Glycogen. Glycogen is stored in there. It's one of the few cells few places in the body, other than the liver, where we have large amounts of glycogen stored. Does anybody remember anything about glycogen? It's a stored energy source, yes. That's a carbohydrate, a complex carbohydrate. It's basically glucose units strung together, and we store them in the liver and the skeletal muscle cells and very, very few places other than the body. So that's an energy store. Skeletal muscles, especially when contracting, consume large amounts of energy. That can be converted to ATP energy. It is converted to ATP energy in the mitochondria. So we see a lot of mitochondria in the sarcoplasm. And of course, that's the powerhouse of the cell. We learned that earlier. That's not a store of energy. It's not energy itself, but it's where energy is made. Just like a power plant is not energy itself, but where it converts, say, coal energy or gas energy into electricity. It converts it into a different kind. In this case, the mitochondria converts energy stored in glucose or glycogen ultimately into ATP energy. Myoglobin. Myoglobin sounds a little bit like hemoglobin, doesn't it? It is a place where oxygen is stored. Many of you might know that hemoglobin is the protein in red. Hemoglobin is a protein in red blood cells that actually stores or transports oxygen. Oxygen binds to it. Well, there's another form of it, at least a molecule very similar in nature, that's called myoglobin. It's what's present in muscle cells. And it tends to bind or store oxygen. And then when muscle cells are heavily using it, needing oxygen, they're heavily being worked, that oxygen is released then to fuel ATP synthesis. But we store some oxygen in our myoglobin. Just like hemoglobin has kind of a bright red color, particularly when oxygen's present in it, myoglobin's kind of a reddish-brown coloration. That's why muscle looks kind of that reddish-brown coloration, is because of the myoglobin. It's basically a pigment that stores or binds oxygen. And of course, all reflecting the incredible energy demands that skeletal muscle has. Myofibrils contain the actual contractile filaments, the contractile proteins. On this diagram, each of these red cylinders is a myofibril. So this whole thing at the bottom is one muscle fiber, that is one muscle cell, while each of these red rods or cylinders here, each one of those is a myofibril, and that contains the proteins that actually contract, where contraction actually occurs.
So are those what grow when you
Yes,
exercise?
yeah, you actually duplicate those or enlarge those, yeah.
Okay. So you do actually gain mass, like
Oh, yeah,
in addition
yeah,
to that.
the cells actually get bigger. You do gain mass, yes. It's just that you don't get more cells, the individual
Okay.
cells get bigger.
So you're getting more material and all. That's where I
Yes,
was
yeah.
confused. Okay, that makes sense.
Sarcoplasmic reticulum. This is a special version of smooth endoplasmic reticulum. All the yellow branching area in here is sarcoplasmic reticulum. What I want you to write down for it now is calcium store. Calcium is going to be very important in determining whether muscle contracts or relaxes. Sometimes it's all stored up inside that endoplasmic reticulum. Sometimes it's released out of that endoplasmic reticulum. It causes different things. But it's a special form of endoplasmic reticulum that serves as a major calcium store. Most of the time, calcium is located inside that sarcoplasmic reticulum. Any questions so far?
What did you say exactly what sarcoplasm was? Like, what do we mean?
Sarcoplasm is the same as cytoplasm. So it's cytoplasm of a skeletal muscle cell, aka skeletal muscle fiber. So again, one of those, I don't know why, other than just muscle fibers have certain physiological properties very different than other cells. They often give special names. Neurons are kind of the same way. Neurons are very special cells. They have unusual physiologic properties. You know, so neurons, the cytoplasm is also called neuroplasm. You don't write this down now. Their plasma membrane is called a neurolemma because they've got a special property of their membranes. Let's think about just a little bit about why we have these striations, this banding in skeletal muscle cells. Remember that skeletal muscle cells have this prominent banding. That's one easy way that we differentiate those from some of the other cells. Let's think about what those bands are. Well, those bands have different names. The dark bands are called A bands, while the light bands are called I bands. So imagine now the whole myofibrils are running this way. So this is showing, think of a big long rod running this way. And you have dark, light, dark, and so on. And we call those dark bands A bands. We call those light or pale bands I bands. How to remember that if you don't remember? There's an A in dark, and there's an I in light. And if you can remember dark and light, an A for dark, an I for light, then you can remember which one is which. I don't think that's how they're, I think they're actually named for light properties, and I don't remember what all of them were. I think A is anisotropic or something like that, but we don't have to worry about those particular naming schemes. Dark and light bands. Okay, oh, here we are, light bands right there. So dark bands and light bands, all alternating. It helps to understand muscle function by understanding that there are these independent or quasi-independent functional units. We call a functional unit a sarcomere in skeletal muscle cells. This one shows a sarcomere ranging from here to here. So it consists of one complete dark band, A band, half of a light band, I band on one side, half of an I band on the other side. What bounds them is something called a Z-disc. It may be hard to imagine this as a disc, but remember you're looking at a rod, a thickened short rod from the side. So if you're looking at a penny, which is a disc from the side, it's going to look like a line, right? So that's what you're looking at right here. This is one sarcomere right here. It goes from the Z disc to the Z disc. And I forget the Z name, it's a German name. Did I write that? You don't need to know this. It's a big long name. Zwischenscheidung. German, apparently. But anyway, I don't have any other way to remember Z disc, it's just what it's called. The end of it's called the Z disc, German word. So thinking about what a sarcomere is, remember it's a short rod. So here is one sarcomere. And these are arranged end to end, successive sarcomeres. There's one sarcomere. If I change this into 3 sarcomeres arranged end to end, it would look like this, where we had one sarcomere here, Z disc to Z disc, second sarcomere here from Z disc to Z disc, third sarcomere from here to here. Well, we can't quite see the end of it, it looks like it's off my screen. That would be the third sarcomere. So another new term, or 2 more new terms we've introduced, sarcomere and Z disc on this one. Okay, let's kind of catch up to where we're at for some of the different terminology here at this point. Then we'll start talking about how muscles actually contract and what those actual dark and light bands are made of. But let's kind of catch up here. First of all, not a clicker question. First one isn't, what is this big pink thing right here? Picture out of your textbook. What is this?
It's a muscle cell.
That is a muscle fiber. That is a muscle cell. So you're looking at one muscle cell here. We can see the sarcolemma on the top. We can see the transverse, aka T-tubules, right here. Let's do some clicker questions now and have you catch up for some of these. Make sure we're all on board here, looking for good percentages of correct responses. Where is oxygen stored? So where would you find oxygen stored at? Which of these choices? Hopefully you can see this. If you're sitting far in the back, you might not quite be able to read some of the words. Okay, and let's see what you chose on this. And you chose mostly D. D is correct, that's myoglobin. So myoglobin is where oxygen is stored. Let's do another one. Where is the most calcium at? Where is the most calcium? Where do you normally find the most calcium? By the way, this shows it in relaxed condition. So this is a relaxed, more or less relaxed skeletal muscle fiber, aka skeletal muscle cell. Let's see where you chose for this one now. Okay, and you chose mostly A. That is correct. Very good. This is the sarcoplasmic reticulum, but here maybe you can see the term. The purple dots, according to our key, are calcium, and most of the time they are mostly in that particular area, particularly in an unrelaxed condition. Let's do another one here. You guys are doing really good on this so far. This is a complex topic, but usually students do pretty good on this when it comes time to the test. It's several weeks away before we get the exam. I think it's the end of the month, so it's a ways away yet before we get to the test. Not real near. Okay, and you guys showed there. B is correct. Okay, that's myofibrils. Very good. That's actually the shorten. That's actually shortens during muscle contraction. Those sarcomeres are going to shorten. That's actually the contraction is going to occur in there. One last question. What serves as an energy store? Oh, there's one last clicker question anyway. I've got one question beyond that. What serves as an energy store? And my little thing on the top just seems to appear and disappear randomly. I have no idea why. Okay. And you guys serve as an energy store. It looks like C and E were kind of split on this one. Let's see what those were. Oops. I think the wrong one. C and E. Energy store. Energy store is primarily E glycogen. Remember, mitochondria is where ATP energy is made, or at least where we convert carbohydrate energy to ATP energy, but where it's stored in is the glycogen. It serves as a fuel source, basically. Mitochondria is just the power plant, which how that's converted to different kind of energy. This shows 3 nuclei. Is that correct, or is that an error on this?
It's correct. It's okay.
Is it correct?
That's correct, yes, because these are multinucleate cells. Remember that skeletal muscle cells are multinucleate. That is correct. Yeah, some kind of cells do have multiple nuclei. We introduced osteoclasts earlier. These are one that have that. Normally, most cells have a single nucleus, but these have multiple ones. Okay, let's get into organization of sarcomeres a bit more and contraction. Again, notice, remember that we've got these dark bands and these light bands. So if you see these dark bands, we have these thick filaments. So look, the dark red line is a thick filament. The thin red line we call a thin filament. So where there are these thicker filaments, A bands, that is dark bands, are created. Note the whole band here, the whole thing is an A band, a dark band. Each of these thick filaments, these thick strands, is what's called a thick filament. Contrasting that with I bands, and where there are I bands, there are only thin filaments. So you can see the thin filaments right in through here, and where you have an I band, you have only thin filaments. Now, some places you have both both thick and thin filaments. That creates dark as well. So as long as there are the thick filaments, it looks dark. You may have additional thin ones, you may not. But as long as the thick filaments are there, it's dark. When there are only thin filaments, then you get the I bands. You get the lighter coloration. Okay, so we have all these strands. Notice the thin bands, the thin filaments. These are connected to the Z disc. The thicker ones are not. These thick and thin ones are made up of different proteins. So here we see some close-ups of the thick and the thin filaments. The top one shows a close-up view, maybe an artist's conception of a thick filament. So, as the name suggests, relatively large diameter, got these little sort of branching heads that sort of project off from it. And it's composed of the protein myosin. So myosin makes up thick filaments. Thin filaments are made up mostly of the protein actin. All these yellow beads here are the protein actin. Now there's some other things involved with them as well. So we've got, in our thin filaments, there's actin, there's troponin, and there's tropomyosin. Actin is what's directly involved in contraction. That's the one that interacts with myosin to create contraction. The other ones, troponin, tropomyosin, in the thin filaments, are regulatory. And by that, I'm trying to have a hard word trying to describe it in just a couple words, but what I mean by that is they help determine whether contraction occurs or not. They're not directly involved with the contraction itself, but their position, their orientation can determine whether contraction occurs or not. So they help regulate that contraction or relaxation phase. But the ones that actually do the contracting is actin. And that's the major part of the thin filaments. Still, lots of terms to start out with. We'll have very few new terms after this for a while, at least not until we get the neuromuscular junction. Now we're kind of getting ready to talk about how contraction actually occurs. Questions so far? Okay, got these myosin filaments, they're thick. We've got the actin filaments that are thin. When people first tried to figure out how contraction works, they looked at a series of pictures, some of them showing muscle in a relaxed state, some of them showing muscle in a partially contracted state, and one of them showing muscle in a completely contracted state. Notice the relative widths of the dark bands and the light bands are changing. So up here, where there is no contraction, there is a lot of light band. The light bands, the I bands are relatively wide. Getting narrower with partially contracted, very narrow here for fully contracted. They thought about this and came up with this idea, this hypothesis or this mechanism of how this works. And what they're saying is, or what the idea for this is that the actin and myosin do not actually shorten themselves. They stay the same length. Instead, they actually slide past one another. And that makes the cylinder, the sarcomere, actually shorter. So, just like my fingers are not actually shortening as I'm sliding them past one another, neither the myosin nor the actin actually shorten themselves. But they do slide past one another, and that makes the sarcomere go from a longer rod to a shorter rod during contraction. It's called the sliding filament hypothesis or mechanism or theory. Pretty well accepted, perhaps we'd call it a theory. Can you figure out what my diagram is showing here? What do you think the red, hopefully nobody's red, green, blue colorblind, I just can't see this. The red here is supposed to be, that's myosin. Here's our thick filaments. And the yellow is, that's the actin, that's the thin filaments. And then these blue over here would be Z-disc. Okay, so imagine I haven't got this colored in the background. Imagine this is one sarcomere. Okay, the thin filaments are anchored to the Z-disc. Let's put this into a contraction cycle. We're going to make it move now. So now we have contraction. This is now a contracted state. We can go backwards if we want to and see that again. Here it is contracting. Notice the myosin hasn't changed in length. The actin hasn't changed in length. It's simply the actin and myosin slid past one another, and that makes the whole sarcomere short. And if all the sarcomeres in a muscle shorten, then the entire muscle shortens. You get contraction, and the body part moves. This is the mechanism of how contraction occurs.
What was the red again?
The red is myosin. So the red is myosin on this. The yellow, the thin, is actin, the thin filaments. And then the blue on the ends are the Z-discs. Let me pause just a minute. Any questions?
Could you explain the sliding filament mechanism?
Yes. So again, what happens? Let's go back to the start here. What's going to happen is the thin and the thick filaments are going to slide past one another. And that's going to bring the two Z-discs close together. That is shortening the sarcomere. But note that the actin filaments do not actually shorten. The myosin filaments do not actually shorten themselves. They just slide past one another. So if we put this into motion, here it is again. They just slide past one another. This is a contracted state. You can see that the overlap is much heavier. This is a relaxed state here. There's not much overlap. This is contracted coming up here. There's a lot of overlap.
So the myosin doesn't move?
I guess it depends on your point of view. No, that'd probably correct. It doesn't move. Yep. I guess well, it depends on how you think about it, but I guess the simplest answer to that would be no, it doesn't move. A little bit more here about some of the parts associated with a sarcomere. How does that return back to the relaxed state? There's going to be energy to actually contract that. How does it return back to the relaxed state? And this is in part due to elasticity of a lot of the skeletal parts. Or sorry, a lot of the parts associated with the skeletal muscular system. There are some actual things called titin filaments, which we've seen here illustrated by a spring-like appearance. I don't know if they're really spring-like, but they're illustrated by a spring-like appearance. They are titin filaments. What happens when you stretch out a spring and then let it go? It's going to go back to its original position. Likewise, if you stretched out a rubber band, elastic item, you stretch out a rubber band, and we basically no longer put force on stretching out, it's going to return back to its original position. Those titin filaments are elastic. And so, or if you compress that spring, like a spring on maybe as serving as a spring shock, on a vehicle. If you compress the spring and you relax, it'll pop back out to its original position. But you stretch it or compress it, either way, it'll return to its original position. a lot of tendons and ligaments, especially tendons, tend to be a little bit elastic as well. You stretch those, they capture some energy, then return back to original position. Kind of like bouncing a rubber ball. You bounce it, it distorts, it recaptures some energy, bounces back up again. There's a lot of elasticity in the muscles, the tendons, the tightened filaments, and that helps to return muscles back to their original position once they've been distorted by contraction or maybe by overstretching, either way. Let's get back to contraction just for a little bit. We've got a bit of a complex figure here, and I'm going to walk you through it, but if you haven't been looking ahead, you'll want to definitely spend some time outside of class looking at this in your textbook. You might want to go through the Wiley Plus. I think there's an audio description of this. Try and explain what's going on. It's a big circle because this can keep going on over and over and over again. This circle describes what happens in a contraction, then a relaxation phase. We're going to start out here with number 1, like they do. And our very start is that we have ATP has already bonded on the myosin heads. So where we're at, this strand is myosin here. These little things are called myosin heads. ATP has previously bound on them, and now that ATP has been hydrolyzed, aka it has been split apart into ADP and phosphate. It's not left the system yet, though. That's basically a way of energizing those myosin heads. When that happens, the myosin heads get energized. Okay, they become hydrolyzed ATP, they become energized. And what's going to happen is then they are going to reach up, they are going to grab and bind onto the actin, and they're going to pull. They're going to almost like ratchet forward and pull. They're going to bind and they're going to move a little bit. And this is now going to create a sliding of those two filaments next to one another. So as the myosin heads bind onto actin, they grab, hold, they bind, the myosin heads ratchet or move forward, and that slides those muscle filaments against one another. Once that happens, the ratcheting happens, or as the ratcheting happens, the old ADP is lost. They're still bound onto the actin. And then what happens is new ATP comes along, and ATP actually causes detachment of those myosin heads. If you don't have myosin head, if you don't have ATP, then those myosin heads doesn't detach, or do not detach. ATP, the binding of that is going to cause them to detach, and then you'll eventually move up in the next cycle again. So ATP, critical step on that, it causes the myosin heads to detach. And then what happens later will automatically follow. Dominique?
What happens in step 3 again?
So step 3, what's going to happen? I think this is showing the pulling. So kind of step 2 to step 3 is kind of a combination of pulling. The myosin actin are sliding past one another, the filaments are sliding past one another. And also at the culmination of this, or at least during this phase here, that ADP that was left on there becomes separated, it becomes lost. It's kind of the transition between 2 and 3, really what both of those things are happening. Got a clicker question here, and this is relevant to something I'm going to talk about here in just a moment, but what happens if ATP is unavailable? So obviously, for a muscle contraction to occur, you can go ahead and be thinking about this one. I'll write something. We're going to need ATP. ATP is one of the 2 things necessary for muscle contraction to occur. If ATP is unavailable, what happens to this particular phase right here? What does it get locked up in? Let's see what you guys picked here. Looks like a little over half of you picked out C, and C is exactly correct. That's a little difficult one to think about. If there is no ATP, Once ATP binds on here, everything is automatic from here on out. Once ATP binds at stage D here, stage 4, it's going to cause detachment, and then the ATP gets hydrolyzed. Once it's hydrolyzed, that energizes the myosin heads. They grab, they bind, they pull, the old ADP is lost. But if there's no ATP, down here is where it ends up with. That is, the myosin heads are still attached and locked onto the actin filaments. Ever heard of rigor mortis before? So what happens when a dead person's in rigor mortis? They're stiff. That's why a dead person is stiff. So this happens generally anywhere from begins anywhere from 30 minutes to an hour or two after death, and then continues maybe for anywhere from another 12 to 24 or 36 hours or so, and the person actually loses the stiffness, loses the rigor mortis. Now, normally what happens in life, we never run out of ATP. This normally would not happen in life. There's something else that regulates when muscles have to stop contracting. But in death, eventually we lose the ATP. ATP is not terribly stable, so after death, ATP either gets used up or it gets degraded fairly rapidly. So what happens is, quickly after death, is ATP is lost. Those myosin and actin heads, or myosin actin, are bound together, they're locked up, and that makes the muscles relatively rigid. Now, eventually, decomposition starts, either by the body's own enzymes or by pathogens, bacteria, whatever, other organisms. They start actually degrading the myofibrils, degrading the myosin actin, and that causes loss of the stiffness, a day or two later. But initially, they're all locked up exactly this way. There's actually something else that's going to be needed for muscle contraction, and this is normally what regulates. We typically do not run out of ATP. Generally, your brain will cause something to shut down, or this other thing will be used up before ATP is used up. So typically, when we get too tired, when a muscle can't contract anymore, when it gets exhausted, that's because your brain says you got to stop, or because there's not enough of this next ion, which is calcium. We're going to learn about calcium's role next in the importance in muscle contraction. So two things for muscle contraction to work. If you have these two present, if calcium is present and available, and ATP is present and available, muscles will contract, and they will keep contracting until something changes. If you do not have both of them, if you just have one and not the other, no contraction occurs. But if both of those are present, you get muscle contraction. And that's going to be our next couple of slides, the importance of calcium. So let's take a look at two scenarios here, with and without calcium. Our first one here is muscles in a relaxed state. So here we're looking at the role of calcium. We're looking at muscles in a relaxed state of relaxation. We now know our pink blob is our muscle fiber. We've got our actin and myosin filaments here, and the yellow is the sarcoplasmic reticulum. The purple dots are calcium. Notice all the purple dots are up here in the sarcoplasmic reticulum. That's where they're normally located. There are some active pumps. That's our little blue thing cycling. It basically scavenges any free calcium that these continually run. They're using ATP energy. They pump calcium back into the sarcoplasmic reticulum. They keep it in there. When that happens, no contraction can occur because this brown rod, tropomyosin, is blocking the binding sites on actin. So you see a key theme or a key idea here. Remember that troponin, tropomyosin are regulatory. They determine whether contraction occurs or not. In this case, they are preventing contraction occurring because they are blocking those sites. Let's contrast this now with contraction. I know I'm moving through this pretty quickly. This is something you're going to want to look in your textbook to review. Let's take a look at what happens during the contraction phase. Similar kind of picture, but what's happened to our purple dots? They have moved from the sarcoplasmic reticulum here out into the sarcoplasm. Various gates have opened up, Diffusion, like they're flooding out. It's like punching a hole in a water balloon. You know, all the water comes flooding out. Or punching a hole in a bag. All the water comes flooding out. So all the calcium comes flooding out. That calcium actually goes down here. Calcium's the purple dots. Maybe you can see that here. These calcium, they come flooding out. They actually bind onto troponin. And that causes a shift in position. This allows myosin to bind to actin. So those who are close enough to see this, the actin binding sites, these little black dots, they're now exposed, and now actin and myosin combine. As long as there's ATP, contraction will begin and will continue as long as this situation remains in effect. As soon as calcium is lost, then the whole system shuts down and you have loss of contraction. So how do we end the contraction? Well, we end the contraction by removing calcium from the sarcoplasm. And we put it back in the sarcoplasmic reticulum. How do we do that? Do you see anything on here that might describe? This little blue thing with an X here, this little symbolic thing, calls it a calcium active transport pump. Basically, that means it's using ATP energy to transport it back against a concentration gradient. And there it's pumping it back in. So once those gates close up, we can transport all that calcium back into the sarcoplasmic reticulum. The calcium is removed, and then what we have is we have the active sites now not exposed. Contraction cannot occur. Okay, we are out of time. I know we haven't, okay, let's make last slide. Let's not do this one. Let's make this our last slide for our quiz right here. So the one on Titan filaments is the last slide. We haven't got enough of the rest of it to really ask a quiz question. We're going to continue with that sarcoplasmic and the calcium next time.